All right, thanks, Dana. All right, our first speaker today is Dr. Julie Urban, and she's a research associate professor with Penn State University. So if you're ready to go, Julie.
Yes, I am, and I presume you can see my screen okay?
Yes, yeah, everything looks good.
All right, great. Thanks, Greg. Thanks, Dana. Thank you all for being here today. I have, if you look on the schedule, 45 minutes allotted to me. So I'll be going through a lot of slides, but I aim to finish here at about 10:30. I have an analog clock I'm watching. And so that we can take questions. And yesterday's conversations and the work that was presented was so good. And I really appreciated the good conversations that came from that. And that's kind of inspired me. You know, this is what I'm showing you today. Kind of, I want to hear what your thoughts are and what your questions are, because definitely from the research end, we benefit from hearing what you're seeing in this. And so certainly the picture in lanternfly land, we have some great results we found to date, but they're depending on how you design your studies, on where you're looking, where you're doing your research, you know, we still see a lot of inconsistencies, and that's just the nature of things. And so what I'm presenting today is about basic biology and how our understanding of that basic biology can better inform our management efforts. And so here, if we look at this, plant hoppers, this lanternfly is weird, okay? It's really different in a lot of ways. And this is kind of, these factors have inspired how I think about it. And so lanternfly life history is really unique among plant hoppers. Any other plant hopper species that anyone knows about has 5 nymphal instars, not just 4. So here we have a really extended, the adult stage is really extended. They have to live for a long time before they start mating and reproducing. And to me, that's an Achilles heel, but it also is what makes them such a problem, okay? And so what we know also about lanternfly that makes it so challenging to manage is that it's highly polyphagous, and what it eats really depends. It depends on what life stage it is. We've talked in depth, and you saw a lot of this yesterday, that its host preferences, you know, are really, really broad at the earlier instars, and then it kind of narrows to the 4th instar in adult. But that's still kind of variable because that really depends on what the plants are in a given area. And that is going to likely change as this thing is moving across the country. And then also what I've been really obsessed about lately is the huge degree of variation we see in wild populations over space. We talked yesterday about how lanternflies are highly patchily distributed. You can have a total infestation 1 block away from where people aren't seeing them at all. And then certainly we're seeing variation, you know, as they move across their different life stages. They move into vineyards and red maples and things late in the late adult season, which is our problem. But also we're seeing kind of unpredictable variation over years in the same places. And so to me, I think of this, we should be getting a better insight as to where and why, you know, SLF is a problem if we understand what does it need in its basic biology. And so today I'm going to talk about 3 different aspects of work that we're conducting in my lab: female reproductive development, feeding, and thermal tolerance. And so each of these, you know, it's basic biology, female reproductive development, and I'll get to why I'm interested in fitness of wild SLF populations in my lab. But basically, to me, what this lets us answer from a very practical perspective is, where can lanternfly live long enough to reach reproductive maturity? We kind of got at this a little bit yesterday, and a lot of what we think about in terms of trying to predict where lanternfly is going to be able to establish, a lot of yesterday's conversations, like I'm thinking of like things that Pierre said about New Hampshire. A lot of it is, where can the eggs survive? And in terms of temperature and exposure, where is there enough warmth for sufficient development? That's a lot of what Melody Keene is doing. And the other aspect of it that kind of relates to what Melody's doing, but certainly how we're thinking about it, not just myself, but also work that Dennis Calvin's done, Brian Walsh and stuff here at Penn State, is with that long adult season, where do you have enough growing degree days? And, you know, where can females live long enough to feed enough to reach reproductive maturity? And so here we know, you've seen the lanternfly life cycle. We have those nymphs that are feeding on a broad range of plants. Fourth start to feed on woody tissues. And here it's that late eggs, that late season adult that I'm really, really focused on. And so some of you, if you've seen me present before, you might have seen me give this part of the presentation, but I still puzzled. I pulled this back up last night and thought about it and rejiggered a little bit of my talk for today based on other things I'm thinking of that I think this study has to tell us. And so in this particular study, this was the lanternflies were collected here across multiple years weekly. And this was a study conducted by Dennis Calvin and John Rost. And basically what this is this is a suburban development outside of Reading. And these little red dots are each of 20 red maple trees that were sampled for lanternfly weekly. And so how they sampled them was that in the earlier season, they put sticky bands on those trees. And you can see sticky bands, we know, can be a problem for bycatch and whatnot, and they're not great for adults, but basically they collected sticky bands and changed them every week and counted the number of nymphs on these bands. And then as adults, John came in and collected the adults into ethanol for me to subsequently dissect. And so he didn't get, couldn't reach every individual bug necessarily on the tree. But these were small red maple trees. Why the study site was chosen is because it was a suburban development. So all the trees were put in at the same time when the development was established. So they were similarly aged. And because of kind of the poor conditions that they were in, like close to sidewalks and things, they generally tend to be a bit smaller trees. So anyway, bugs were collected out every week. And so they started first, what did they see? First, egg counts were done. And so here, if we go, these were their weekly counts from what they found in 2019. And so at the egg stage, if you can see my cursor, what they're finding is an average of 475 bugs per tree. So they counted egg masses, and we know that there's 30 to 60 eggs per mass. And so when we come up with these estimates, we use a conservative estimate of 37 bugs per egg mass. So they counted their egg masses times 37, and that gives them an average of 475 bugs per tree. And then as you put the bands on, you can see that, okay, those first instars, they caught an average of, there were 250 bugs or so per tree. Second instar, a bit fewer. Third and fourth, progressively fewer. Two things to note. One is that, okay, this looks like the traps are working. If you're putting traps in, these sticky bands, and you're trapping bugs out, removing them, killing them every week, that it looks like you're reducing your population. The second thing, thinking about what we heard yesterday, and it's not that any one study is wrong, it's just we need to look across studies to understand the big picture. Yeah, they don't like red maple during the early instars, but that doesn't mean some aren't still there, right? They're still finding them, average of 34 a tree, at third instars, 17, so they're still around. Also, they're likely moving through a baby, just like inventively feeding on maple. And so, it's not that they're never feeding on maple during these other life stages, they're feeding on a lot of stuff. So, okay, what happens then when the adults come in? The adults come in force. Now you have them. Clearly, they're moving in from a different area. How far away? No idea. There you have an average of now 600 bugs per tree. Again, those are removed every week. And what happens then when eggs are laid? You kind of would think if you have somebody collecting them out every week, you're going to get a reduction in eggs. So eggs are laid, count the eggs, multiply times 37. How many bugs are there then in the egg stage? Doink. You have to change the scale now. It's the same thing. Look, you still see your 615 down here from the adults. We have to change the y-axis because we have now, with the egg masses, an average of over 35 bugs per tree. Okay, so that in and of itself is interesting. So certainly they were moving in, probably from a distance into that housing association. So you look at this, how far were they coming in? That's a question we'll get to here with my data. And so basically what I do is I'm interested in, as these females are coming in, are they mated? And so you saw yellow bellies yesterday. And so what we do in my lab, and my technician, Elena Gomez, is a wonderful help with this. She's working on some cool stuff I'll share in a minute. But basically, we image the insects and take that yellow area above the spiracles and measure it. And then I've been doing dissections. And so I look to see if they're mated. And this is what the male gives to the female and what it looks like once it's inside the female. That's the spermatophore that contains the sperm. And I rate how reproductively mature the females are there eggs that are present, and then relate that to the size of that yellow region. And so at first I was interested in this in terms of growing degree days. And so I have growing degree day, how I computed these things was based on, the literature, what threshold I used. And so here what I'm showing with you is that we have 2 years of data. 2019, the collection dates were a little bit different than they were in 2020. They were collected a little bit earlier for me in 2020, but we have, again, adults were collected off of the trees and removed from September to November 15th. And so what does this look like in terms of yellow belly? So if you look at yellow bellies here, you can see that, okay, they start pretty small and they get progressively larger over the season. And so here, as I do the dissections, the first spermatophore I find is from September 25th. Degree days was like around almost 1900. By the next week, nearly everybody's mated. And that's when I see the first mature eggs. All right. And then by beginning of October, nearly everybody's got mature eggs. And so a couple different lessons here. And one of them that really stands out to me from hearing the talks yesterday, and especially if you've seen me present this before, please note, I've never said this before. The first eggs that were laid in the field were observed to be laid September 25th. And the bellies were little. Look at that. The average size where we see reproductively mature females and the first really mature eggs, and egg laying happens here in this year. That last week of September is when it began. Those yellow bellies were relatively small compared to how big they get. And so I don't know how people are estimating these things, but I know I've heard folks say, when they intercept a female, no, she wasn't reproductively mature because she wasn't big and fat. They can kind of be small when they're reproductively mature. This is why I'm doing this work, to really figure that out. And so there's also other studies kind of coming out of Penn State that I was involved with that did flight tests of females, and those early studies that did those, controlled dispersal flights found that the females that were flying far weren't mated. The females that weren't really flying very far in that test were mated. I think that, the conclusion of that study that, don't worry when females are coming in, when they're exhibiting these big flight bouts, they're not mated. No, that's not true. That's not true at all. We're finding little ones mated, and these bugs are removed every week from these trees. So the fact that more keep coming in when they're big and fat means, yeah, they're flying when they're big and fat and mated. So SLF is likely to be mated and gravid when they move into new areas. So here, the first observation, the first egg masses at this site were seen in September. And then the first egg masses were seen at the same site, same week in September the following year. But what was really interesting was that 2020 was a lot cooler in terms of cumulative growing degree days. And a lot of what, I think what came up yesterday, I know Melody Keena said it, is that we think that egg laying is also, certainly driven by development, but also there's other cues like day length that seem to be involved here. And so if we look at growing degree days between 2019 and 2020, again, I have bugs collected from the same exact trees. We see that 2020 is a cooler year in terms of having almost 250 fewer degree days. And there's all sorts of ways that, these cumulative degree days are, driven, and Dennis Calvin's working on a paper that has taking a really cool, complex approach to dealing with this. But basically, my prediction here is that even if egg laying is triggered by another cue like day length, we would really expect that if development is driven by growing degree days, which to some extent it is, then we would expect that in 2020, the development of bugs would lag behind that in 2019. So looking at that yellow belly size as my proxy for development, right? It's highly correlated with weight. I didn't show you that slide, but basically 2020 development, yellow belly size should lag behind that of 2019. And so while I'm still doing the dissections for 2020, we have the data for the area from over 400, almost 450 females that we imaged, that Elena imaged. And so here's the yellow belly size for that cooler year 2020. And when I overlay the warmer year 2019, is it the case that it lagged? No, not at all. Belly's got if anything, belly's got a little bit fatter earlier in the cooler year. So not at all what we would expect, okay? And so is this unique to the site? Certainly I have samples from other places. I'm looking at, are they getting cues that it's a cooler season from the host plant so that they speed up a little bit? Or is it the case that in the scheme of things, this isn't a big deal in terms of degree day difference between these 2 years? That's what I'm continuing to ask. What is interesting here is that when I look at yellow belly size, back. For field collected specimens that we have versus, and this isn't from all studies, this is just from a handful of specimens reared in labs, what you see is that there's not only wild populations tend to get much bigger in terms of yellow belly size versus lab reared specimens. But also I would like to look at more lab specimens. So like Tracy Lessie's doing a great job growing these things in the lab, and she's getting them to lay eggs. And so hers are probably even a little bit better than these. But basically, I have been looking at this, thinking about it's so important to assess the variation in wild populations. And part of what I'm thinking there, and I'll show you what we're doing along those lines now, part of what I'm worried about there is that if you can get that big of a yellow belly, right, and they're laying or some are at least beginning to lay when they're smaller, it appears to me, because they can lay subsequent egg masses, if you have a longer growing season as you move south, our population is going to get that much bigger because they can lay multiple egg masses. But the other side of things, if you look at these are kind of lab reared specimens, under not great conditions. I think a lot of lab reared specimens, like what Tracy's doing, has more fit ones that are going to be bigger than these. But basically, the question of, if we're seeing in the field 4 millimeter size bellies when egg laying begins, this raises the question of how small, or essentially almost even nutrient deprived, can they be and still lay eggs? That might inform that northern limit for them, okay? So anyway, what we're doing there along these lines is Elena Gomez she identified field sites, and basically what she did is, starting in May, I want to get a sense of the variation in these wild populations. So we identified a bunch of sites, and 1 week she would drive from State College, which is about here, all the way to New Jersey and sample 20 bugs per site. These are the sites that kept enough lanternfly for us to do this for the whole season. And then the other week she would drive west and collect Pittsburgh. So each of these sites had bugs collected biweekly. And when she brought them back, she measured the live mass and froze them. And we're going to analyze their carbohydrate levels, protein levels, and lipid levels to try to come up with this sense of nutrient status and fitness of wild populations. And so we haven't done the nutrient analysis so far, but if we look at just here, the mass, here's the mass of first through fourth instars across the season, and then of males and females. And a couple things to notice here. Cumulative growing degree days here, it looks a little bit different. This is in Fahrenheit, not Celsius, so it's not really comparable. So just to give you kind of a sense, egg laying, that's about the end of September, 1st of October. That's where egg laying started. And so what really stands out to me is how incredibly variable those sizes are, right? And so what does a female need to lay? How big does she have to be able to lay at all? And if she's big, how many clutches can she lay? That's what we're looking at. And again, if we look at just the adults, you get the sense of, wow, there's just a huge degree of variation out there. And certainly a lot of that is driven by just individual differences, but also in terms of like the relative nutrition that they're getting from their host plants. So we're going to continue to look at this and try to relate this to basically potentially down the line what they're feeding on, to the extent that we have older infestations that have more crowded populations of bugs versus like that moving front. Okay. So to go on kind of the next thing, I wanted to spend the most time on that because I think you might have the most questions on that. Some of the work that we've done that I've presented elsewhere is looking at host plant suitability. And this gets at the question, what other specialty crops are at risk of feeding and possible damage? Scott, you mentioned Illinois yesterday being worried about horseradish. That was a new one to me. That was really cool. But we've kind of looked at this, and this is the work of Holly Schugert, a postdoc in my lab. It was started by Heather Leach, but Holly's done a lot of it. And so here, in order for a different crop to be suitable, first of all, lanternfly have to be able to survive if they feed on it. So essentially what we're doing is force feeding lanternfly a bunch of different crops. And then where we can measure to see if there's some feeding damage. And here, I'm just going to click through these slides very quickly because I have summaries at the end that are more big picture. But I do want these to be available on the recording, and then we can come back and in my question time, we can ask about certain things. But just to show you, this is the variety of crops we've looked at. We started with adults because they're the heaviest feeders. They're going to potentially do the most damage, then fourth instars, and then we've done a little bit with first through third instars. And because we're just doing this kind of as a screening to identify some plants, then some plant species that we want to look in more depth at, we're limited in terms of the number of plants we can do. So our sample sizes are really small. But what we see overall is that a lot of these things, nymphs can feed on all sorts of stuff. And so, yes, they like to feed on cucumber, and certainly that could have impacts, at least in the limited stuff we looked at. Heather here showed it did have impacts on yield. Also on blueberry. The earlier instars do better as they get older. They don't do as great. Same thing with strawberry. They like to feed on strawberry, don't do as well later on. That's also because these plants are exposed. It's hot. They really don't have anywhere to hide. Here with pumpkin, they can survive longer on pumpkin. They do great on pumpkin, even if they're with, if you're in a bag with 24 other or 49 other nymphs, you're still surviving pretty well, even into June. And this is likely because in pumpkin plants, these are bigger leaves. If it's hot, they're a little bit less exposed. They can hide underneath the surface of the leaves. So temperature is kind of important here. When we pull all these things together, and we looked at, in this case, comparisons of how did nymphs do when we put them on strawberry compared to blueberry, hops, pumpkin, tomato, and sunflower. They did best on hops. And they overall, if you look at these transition points, the star, the triangle, they molted and developed a little bit faster on hops. This begs the question, how long, if you put them on a plant, if you left them without food, how long would they live? And this is certainly important for their potential transport. And so here, how long they'll live, about 3 days, but that really depends on temperature. I'm going to do that a little bit more in the lab. And then here with hops, a lot more interest. We looked at adults as well, because I'm really worried about things that are good hosts for nymphs and adults. And so you can see when we jam 100 insects onto hops, it can really do some damage. And so we did some further work with that. What we found in the back row here, these are the plants. Some of them were heavily fed upon the year before and were heavily damaged, but they really seem to be resilient. And so we're very interested in further work on hops, really loading on a bunch of nymphs. They survive well as nymphs is kind of the point. They survive well as early season adults on hops. They even seem to do well later season on hops. Certainly when you jam that many bugs onto the plants, it can do a number. So we're continuing to do some work on hops, but also kiwi fruit is another one of interest with collaborations we have with folks in New Zealand. And so here We have nymphal and adult survival looks kind of good even on kiwi fruit. And so here we're going to continue to do that. We're looking at how nymphs do on kiwi fruit. But to me, hops and kiwi fruit in particular are problematic. I was just in New Zealand, and so certainly kiwi is of interest to folks in New Zealand because given that we're seeing adults do well on it, that's a concern because adult feeding and presence on kiwi means that the adults would be there producing honeydew when that could impact the kiwi fruit. There's a different plant hopper, the passion vine hopper, that feeds on kiwi fruit later, and by kiwi fruit, I mean the plant, later in the season as adults. And their honeydew is very sticky. It basically is a problem for sooty mold growth on the actual fruit of these plants, on the kiwis themselves. And so the kiwi fruit skin is sticky. And so sooty mold can grow on it really easily. I'm concerned about kiwi fruit and hops because, again, hops could be sensitive to honeydew. And so things like that, where you have nymphs and adults feeding on them, are a concern to me. So we've come up with a risk assessment here, and we're continuing to add to this. But overall, when we're looking at nymphs, yeah, it might be a problem, but really, and nymphs can survive really well, but it doesn't look like there's much damage. Fourth instars, you kind of get a little bit more of a problem, again, likely because they're feeding more heavily. But here, adults in particular is where we see the heaviest damage. And so we can talk about if you have any questions with this, but I want to add one more section here. I have a couple more minutes. Thermal tolerance. And so here, this is different than the work that Melody Keene is doing. She's looking at different temperatures and climatic regimes and how that influences development across the life stage. I'm just interested in my lab, we're just interested in exposure. If nymphs at first instars are exposed to extreme cold, or if adults are exposed to extreme hot, what are those thresholds that SLF cannot survive, because this has implications when they're transported. And so some preliminary stuff real quick. This is my master student, Liz Deecher. There's Liz down there. And she's looking at cold tolerance of first instar nymphs. And so here I just want to give you a couple results that she has, but if you're interested, she'll be presenting more of this at the Eastern branch meetings for the researchers here. So what we do is we have, the first thing we're interested in is, when do they pass out? That's called CT min, or chill coma. So this is what's kind of the warmest temperature at which the insect loses the ability to move. And so this is an important, like, kind of first threshold to report in the literature. This is when they pass out and it might look like they're dead. And so what we do is, this had 10 first instars per replication, for 5 replication. So we have this circulating water bath, and we started it out at 15 degrees Celsius. So that's 59 degrees Fahrenheit. And we put the bugs in, and this is just them in a test tube with some mesh over the top so they can't jump out. And here we're essentially letting them acclimate. And so the temperature was lowered 2.5 degrees every 2 minutes until we reached somewhat cool temperature of 7.5. And then we would pull them out, do a tap test, tap it, see if there's any movement at all. Have they passed out yet or not? And then at that point, we started lowering them by 0.1 Celsius. And then they'd be held there for 2 minutes, allowed to acclimate, tap, tap, tap. Are you still moving? Lower it more. So that's what we did. And then we recorded when each bug fell into a chill coma. And so what we found was that we measured both the temperature of the bug by putting a thermocouple in, and recorded the temperature of the setting. So basically we see that they pass out kind of at what I think is a warm temperature, 46 degrees Fahrenheit. Their temperature was 7.6. It was when the bath was set to 7.3. And so they look like they're dead at that point. It took them really, on the average, less than 2 minutes to recover, and all fully recovered. And so that was our first pass here. But then the real interesting thing is lower lethal temperature. So what's their survival when they're exposed to a constant temperature for a constant time interval? So we're not letting them acclimate everybody in the pool at a cold temperature. And so here, what I'm sharing with you today is just 3 reps, so 10 bugs per rep. And so here, the circulating water bath is held at a constant temperature. And you put them in and leave them in for an hour. and then see what the survival is. Okay, so basically, Liz observed them constantly for up to 2 hours to see if they recovered. We were concerned any longer after that, they might be at risk of starvation. So we just set the limit of our observation at 2 hours. And so here we started at that chill coma temperature of 7.5. And again, this is a different set of bugs, each exposed for an hour. And so here, this is the last of what I have to present to you today. I think this is cool. Okay, did they recover? What was their survival? Okay, do you want to take bets here? I would bet, you would bet, right, with that chill coma, what we found there, you'd expect when you expose them to 7.5 Celsius for an hour, yeah, these little suckers are going to survive. Okay, but what about colder? And again, we're not allowing any acclimation. What about 5 degrees? 100% survival still. What about 2.5? We're getting awfully cold. Now we're thinking if these are in a refrigerated truck and they're left in there for an hour, are they going to come back or not? Yes, we had a little death, not much. Okay, what about a freezer truck? If you stick a bug in a freezer truck for an hour, are you safe? All right, started to see some death there. That's kind of promising. What about minus 2.5? Certainly that would kill them, you would think. No variation, no, not so much. So here, Liz has done some subsequent tests exposing them to -5 and -7.5 degrees. She's also done work where you test exposure times, putting them in at -2.5 Celsius from 60 to 330 minutes, kind of estimating how long would be long enough to kill them if they were in a shipment. And so anyway, with that kind of next steps, we're continuing to look at more bug sites, you know, trying to figure out northern and southern limits and looking at that nutrient content. But basically here, where I'm at is I have the rest of the time now for questions and ideas with what you all might want to know or have suggestions as to other things we could look at. Thank you. Greg, I'll turn it back over to you to moderate here.
All right, thanks, Julie. Yeah, there is like as expected, there's quite a few questions for you
Cool.
in the Q&A. So you probably have to answer most of them offline.
Okay.
Or, you know, I mean, not live, but yeah, I could just read out a couple of them to you. So how does your analysis change your opinion about how far north they might be able to establish?
I'm worried. I'm honestly worried. And this is something like up until this point, you know, I think that like the best predictors, and I don't know if our Dean Rick Roush is on, but a lot of the conversations we've had in-house have been that Dennis, and I think with you all, you know, Greg and Matt Travis at APHIS, we've said that so far Dennis Calvin's maps, looking at growing degree days and where there's enough time for females to reproductively develop, has been the best predictor of where they really have seemed to be able to establish. Like we haven't, you know, within Pennsylvania, except what Brian pointed out after looking at my data here with the slightly colder region, Dennis's work suggests that they shouldn't be doing as well as they are, like in Scranton and in Northeastern Pennsylvania. And so this idea that, you know, maybe you don't have to be as you know, how many eggs do you need to lay for a population to still continue to expand and be established farther north? It might be a little bit more permissive than I at least was thinking up until this point.
Okay. And how long does the spermatophores last in the female?
It seems to last quite a long time in that I'm finding them across that whole, I haven't really done anything to watch the degradation of any one. I'm getting snapshots of individual females, so I can't answer that completely fully, but basically by the time they're in the female and in the structure they're in, they're rather crystallized, and so I see remnants of spermatophores at females of different ages. I see evidence of multiple matings in terms of those little wings that are there. And so I basically, our understanding, and again, this isn't really written about, but kind of how it works with other insects and what seems to be the case by looking at these guys, is that the spermatophore gets passed, it becomes more crystallized in the females. As the egg then passes, when she goes to lay it, that's when it's fertilized. And so likely, they can be in there long enough for multiple spermatophores to be together at the same time. So likely multiple parentage in the same egg mass from what I'm seeing.
Okay. And there was one comment question about spotted lanternflies appearing to be smaller on average in 2022 than prior years. One person speculated it might have something to do with the drought conditions last year. Is it possible that drought conditions affect the pressure of sap within the tree enough to inhibit feeding activities?
Yes, certainly. And I would also think even the presence of other insects at high densities would do the same thing. That's why we're looking at mass, and that's why we're looking at the nutrient status that we are, and why we're selecting sites at different areas of insect densities. And we plan to do this. I think Elena will probably move on in her career to other things, but I'm going to continue to do this in my lab for multiple years so that we can kind of hone in on that a little bit. In talking with Elena and with my postdoc Holly, also what we might, we want to look at these data we've collected this year once we get those nutrient analyses in it, kind of as just like baseline things to inform what other studies we want to do. But if we want to connect that to trying to tap into some of the phloem in these trees, if we can, and kind of relate it to that. Yeah, I think that's exactly where we want to go with this, because that's what we're wondering too.
Okay. Yeah, because that was a thought that crossed my mind between the 2019 the 2 years you compare, the 2019 and 2020.
Yeah.
2020 being cooler, that possibly that the sap flow is better in the cooler year, so that compensated even though it was cooler.
Oh yeah. Oh, cool.
And then is hemp a host plant of SLF, since it's the same botanical family as hops?
We haven't looked at it, but we've talked about looking at it. And so I think that would be very interesting to do. I would think so, but I don't know that anybody's tested it. We haven't.
Okay. Have you determined the amount of time at freezing it takes to kill them, or is that work to be done?
You mean in terms of what temperatures does it get to freeze them?
That's what I'm guessing.
Yes, that's I don't want to present any of data that Liz hasn't already presented because it's her master's thesis, so I want to present it. But that's yes, she's getting to that by looking at the 1 hour long exposure at lower temperatures and the minus 2.5 for longer time periods. Yes, we will get at that, and we're starting to get at that with what we've done so far.
Okay. And have you observed direct feeding on hops cones?
Oh yeah. Oh, on the cones. No. We've seen ambient, and Holly, I don't know if Holly is on, if Holly wants to write something in the chat if she's seen it, but no, my understanding is not that at all. But we have seen what we call ambient lanternfly, so where we have the hops put in at the Penn State Berks campus. Holly and Elena had to pick off like wild lanternfly that were already there feeding on those hops on the vines before we started our study. So they are interesting to lanternfly in the area as plants, but not the actual cones.
And should we expect to see SLF hatching sooner in spring after warm winters like this one?
I mean, I would defer that question to more Melody Keena's expertise, but I think that there's so much variation, there in any of the life stages, but certainly with hatch, there's a huge amount of variation. And so I think that a lot of that variation is individual differences. Some of it might be driven by whatever heat exposure they might have gotten in the fall, or where we were not really measuring it, but also even microclimates in the same area are going to make a difference. And so I don't know that there would be a strong enough signal just from that we would notice it with that amount of variation.
Okay. And if day length turns out to be a factor in reaching reproductive maturity, do you expect latitude to be a major limiting factor in northern range limitations?
Yes, but there,
Because I know there's many factors that go into, right?
yeah,
Whether, yeah.
yeah. So but not again, any one thing isn't necessarily a deal breaker. Yeah, because just as Greg, you said, there's a lot of things that go into it. So I'd be hesitant to say.
Okay. And there's quite a few more in there. I just covered some of them, Julie. So yeah, and I'm sure there'll probably be more popping up. So if you wouldn't mind going in and answering some of the questions.
Sure thing. Thanks, everybody.
Yeah, thanks, Julie. All right, so we can go on to our next speaker. I see Matt's getting everything queued up. So our next speaker will be Dr. Matthew Helmus. He's assistant professor with Temple University. So whenever you're ready, Matt.
Great, thank you so much, Greg. So today I'm going to talk to you about our work on forecasting population dynamics in spotted lanternfly, and we're going to focus a lot on growth and dispersal. And when we consider spotted lanternfly and growth and dispersal, then of course we're also considering how humans are affecting the growth and dispersal of lanternfly by their establishment and their transport potentials. And so our team at Temple University thinks a lot about sort of how we manage this pest in an area that isn't necessarily on the front lines of this invasion, but is in the core. And so just in the last 10 years post-invasion, these are some of the quotes that I've heard from some of the wine grape growers. And I know that many of you know this already. We've lost about 20% of our vines to spotted lanternfly. The population of spotted lanternfly in the vineyard has increased every year since 2017. This is, in the core area. Spotted lanternfly remains one of the most limiting ecological constraints to sustainability of our winery. And the real challenge is the invasion that invariably comes in September when they invade in waves from the surrounding areas. And so it's in this context that we try to approach all of our work on forecasting population dynamics, not just at the broad scale, but at the small scale too. And so Dr. Jocelyn Bem works mostly in field biology. I work mostly in data science, and Benjamin Seibel works mostly in mathematical modeling. So the research I'm going to talk to you about today comes from our three labs that combined, we think, or argue, is the best way to think about how to forecast or model spotted lanternfly population dynamics by merging these different fields. And there's certainly other fields as well that should be merged to help with forecasting. So today I'm going to talk to you about three of our research objectives. I'm going to spend a lot of time on how we forecast the establishment of spotted lanternfly. And so under this objective, one of the first things we had to do was build a database. So there is no central database of spotted lanternfly occurrence records. We've all seen maps. Different agencies collect data differently. Agencies have different memoranda of understanding to actually gain access to this data. The abundance data is collected across agencies. It's not standardized. Public reporting tools vary across states, et cetera. We're all collecting data a little bit differently. And to really get a good picture of where lanternfly has been and where it's going, we need to get all those data together in one place so that we can all use it. And that's what our postdoc, Seba de Bona, has done. He created an R package, it's our data package that brings together all these different data sets, harmonizes it, and then provides it. And so what we're doing with this database is we're working with agencies, researchers, et cetera, to gather these data sets that are being produced. We then process that data, we harmonize it, we standardize it, and of course, then check for any errors. We then take those data sets and it goes through a strong anonymization process where different properties, different exact coordinates aren't shared, but they're all rasterized to a 1 kilometer grid. And by doing that, then this data can be made open access so other people can actually use it. And so then we distribute this data as an R package, but it's just a downloadable data tables and maps and data summaries that can be used by anyone, even if they aren't necessarily using these data in any coding application. So this is one of the images of the data set that's available now. This is at the 1 kilometer scale. This is from 2021 to 2014. You could see that invasion core area where we think about what's happening with lanternfly now in that invasion core area where it's completely naturalized. And in this data set, we also have estimates of the density per kilometer squared, low, medium, and high. That's the best range that we can get from all the data. And then on top of our data sets, we build open access maps and apps. And so this is one of the apps that's associated with the data set. You can look at the density through time, the county scale, or at a slightly smaller scale at the 10 kilometer squared scale. And you can take a look at how it's spread here in areas across the United States. And all these data, like I said, are available. And then we use these data then as well to try and estimate how long ago spotted lanternfly invaded a particular location. We know that lanternfly is really patchy in the landscape. And so by using all this data and running an interpolation model, then we can find areas that maybe are slightly more, have been invaded slightly sooner rather than later. And so we have an estimate of age for different locations and sites as to when lanternfly invaded that particular location. So this is a picture of Victoria Ramirez. She was a fantastic undergraduate who just, I think yesterday it came out, published her paper in the Journal of Applied Ecology. And what she did was she took this data science estimate of site age. And by site age, I mean, how long ago did lanternfly invade it? And then looked at egg masses and looked at how multiple spatial scales can influence whether or not you're finding different egg masses. And so by sampling multiple sites across the invasion core to invasion edge at these different ages, what we found was that if you're in the field looking for egg masses, that you have to think about how long ago that site was invaded by lanternfly. So at younger sites at the invasion edge, it seems like, based on our analysis, that managers should expect to find really patchy colonization of sites. But as soon as you find a site, then that tree is going to those trees are going to have lots and lots of egg masses. And so it's that at that point, then, if you focus all of your efforts to try and eliminate that site, that could actually do a lot of good in slowing the spread. At older sites in the invasion core, they're more likely to always have egg masses present, yet often at a lower density. And so it makes them really difficult to identify on individual trees. So you have to take different approaches depending on how far along in the invasion you are at any particular property. I'll pause here just for a second. I'm not going to go through this figure, but this is basically summarizes all the analyses. And those of you at home who are watching this and can get the video, you can pause this. So please contribute to this open data project of ours. Researchers, your work gets more citations. If you include your data, we articulate that your work should be cited first before citing our data. We are aggregating data is what we're doing. So please contribute. And state agencies, please contribute your data. You get clean data that your team can work with, and then of course you get robust model estimates. So please email me if you'd like to contribute. So I'll go through sort of a list of these deliverables. So here, one of our deliverables that we've produced at Temple University is an open access database for spotted lanternfly modeling. So let's focus a bit on our actual modeling using those data. And so we've done some statistical niche modeling. And one of the first models that we built was based on a niche model. We used this to machine learning algorithm called Maxent. It relates the species occurrences to environmental covariates. And so we built lots of different models based on temperature, precipitation, human development, et cetera. This is an estimate then of where lanternfly is likely to establish across the United States. And we've seen maps like this before. This is a very fine scale map that we were able to produce. Like the other data sets, these maps and data sets are available as well as our apps. And so you can take a look at different areas of the country on some of our apps. You can vary the strength of that establishment potential. So how well lanternfly should be doing in these different areas. And you can take a look at your landscape, and decide whether or not it makes sense for you to survey that landscape or not. And you can zoom in different areas and also take a look at where on Earth are the important wine growing regions. And of course, they correspond strongly with lanternfly. And then at a finer scale, you can also take a look at where in the United States are the grapes grown and what that establishment potential is, where lanternfly will do just fine if they get there, based on this particular model. But we know with these different estimates of establishment that they're highly dependent on global change. They're highly dependent on how the climate's going to change in the future. Sam Owens is one of my new master's students, and what he's doing is asking this question of, based on the statistical modeling that we can do, how will climate change affect establishment potential? So he gathered lots of different data sets together, selected variables that didn't have a lot of collinearity, trained these different machine learning models, and then made projections into the future. And we're just doing out to 2040, so not something far off in the future. Based on the modeling, this is lanternfly's historic range. This is sort of the range that we think about, that lanternfly can get to now and can establish. Orange is the predicted range, and the points are where spotted lanternfly was in 2021. And so by doing then the modeling, we can then estimate, just in our 2040 forecast, what we expect climate change to do. And you can see there's a massive increase. That's because climate change is occurring so rapidly and is really expanding then the available temporal niche. And so you can see areas in that previous map, like in the Upper Peninsula, that could start seeing lanternfly. And you can see this at a finer scale as well. This is just New York. And so you can see sort of how these areas will increasingly make it more likely for lanternflies to survive. And so we are already dealing with this climate change now. We're dealing with the effects of climate change, and we will be even more in the future. And so from these statistical models, then we have global establishment risks of establishment for lanternfly. So besides statistical models, we're also developing first principles population dynamics models. And here we're focusing on temperature-dependent growth, because temperature-dependent growth is the most well-researched aspect of lanternfly biology. Stephanie Lukowitz, she is a postdoc in our group, and she's modeled the spotted lanternfly life cycle from egg all the way to egg-laying adult. And so these population models that we build, they tell us how the total number of individuals in a population changes over some period of time. And if we can assume the population is large, change continues, so we can average things out, then we can use differential equations that are based on mortality, development, dispersal, et cetera. For spotted lanternfly, we're to a point with existing data that we can pretty well model how temperature should affect the mortality and development of lanternfly. And so that's what we're focusing on in our current models. So essentially, we're taking temperature-dependent growth experiments, such as those depicted in this graph here. We're parameterizing those those data from those growth experiments, then are used to parameterize our system of partial differential equations of the spotted lanternfly life cycle in order to look at population dynamics. And so what we found with these population dynamics models is that depending on where you are in the country, population dynamics of lanternfly can look very, very different. So here we have Philadelphia, and here we have LA. So in the top, we have eggs moving through the system, and then we have lanternfly developing here through first instar to egg layer. In LA, we have this group of lanternfly moving through the system, and then we have a second group moving through the system, all within the same year. And so what we have in LA versus in Philadelphia is the potential for lanternfly to have multiple generations, and those multiple generations can either have diaposing eggs or non-diaposing eggs. We don't know if this is true, if the temperature profile in LA will actually allow lanternfly to have, multiple generations. But this modeling exercise essentially shows us a known unknown then that we need to understand. And so we can take these population dynamics models and then calculate an R naught value that says where in the country is that R naught value above 1. And so where in the country might lanternfly be able to establish? And so let's dig into a little bit of the math here. In a lot of the talks I've given, I haven't really gone through the math too much. I'm not going to go through the math here, but I'm going to illustrate how we use experimental data, a lot of the data that Julie talked about previously and others have talked about. We use this experimental data then to parameterize our models. And so the basic model that I'm showing you right now is a model where we're modeling the size of the non-egg population. So these are instars and adults. These are the motiles. These are the individuals that we see running around as a function of development rate and a mortality or per capita death rate. And so development rate is a function of temperature. And so it's how fast they develop per day. And that development then is a function of the temperature of that day. And so that's the standard developmental degree day functional form that we've seen, we've heard of. This work comes from work, at Wyomissing that Julie just talked about, et cetera. And so these are these degree day functions. Mortality rate then comes from other experimental data that you've seen on temperature dependent mortality. And that has particular functional form as well. So what are these functional forms? So these functional forms are termed biological performance curves in general. So this here is like a thermal performance curve where you're measuring performance of an organism in some way and how that increases then decreases as temperature changes. And so there's some thermal tolerance breadth or width. And so these types of curves then are derived from first principles that are parameterized then from data provided by field or lab biology. And so we have some sort of theoretical idea of what the function should look like. And then we're parameterizing that function with data. So here are some of the functions that underlie the population dynamics model that I showed you the videos of and the R naught values for. I'm not going to go into all these. I'm just going to focus on one, just this panel C. And this panel C has the 3 different functional forms for egg mortality rate. So we have diapause, post-diapause, and non-diapause eggs, and we also have motiles that are on here as well. So these are those other individuals that have already hatched. And we're going to focus just on that yellow line there, the diapausing and post-diapausing functional form. I'm going to step through how we calculated that. And so when we did this work, we found 2 studies that we can use. One was Park 2015. What they did was they took eggs, they collected from the field, they kept them at different temperatures at -15, -10, -5 for a certain number of days, and they measured mortality. And so you can see the data here for those different experiments. And then another study, Keena and Nielsen, 2021, estimated egg survivalship, egg mortality at 5 degrees and 10 degrees. So they held these eggs at these constant temperatures. So for each one of these different data sets then, we can estimate what the influence of temperature, time is on mortality or survival. And so we can fit curves then, and those estimates then allow us to build these functional forms, those beta values of these exponential decay functions. So in the end, what we get from all of this, because we had one data set that wasn't working, so we had to throw it out. We ended up getting 4 data points then that allows us to directly relate egg mortality to temperature. And so these are the observed data. And then the functional form that we ended up fitting was this simple breakpoint line, where once you start to get below -5, eggs start to die. But above -5, eggs don't die at all. And then we're able then to produce these maps based on the functional forms that I talked to you about, as well as this egg mortality one that I showed you. These are not maps. So we can also make these maps different in different color schemes, different aesthetics. And so this is another one to sort of help us see how these maps are changing. So anything that's blue then has an R naught value that's less than 1, which means that they can't establish. So we fit this breakpoint functional form. It made sense based on the statistics of the point at -5. There's really high measurement error there. And so we didn't want to really anchor our functional form there. And so we made this simplifying assumption that this is what the functional form looks like. But there's only 4 data points here, these 4 red dots. And so in reality, you can essentially fit any type of function to 4 points. There's not a lot of data there. We did the single breakpoint. We thought that made the most sense. There's a double breakpoint that we could fit. That's panel D over there. And even the exponential in C that we could have fit here. And that exponential actually better explains what we see in the thermal tolerance curve here, sort of that smoothing that we see here. And so how then does an exponential do? And what we find is that these differences in these functional forms, they do not fit the spatial data very well. That single breakpoint fits the spatial data. We know where it is, but those exponential fits and the double breakpoints do not fit the spatial data at all. And so even though we can fit these different models, and the statistics of fitting the different models all say that all these models are equally valid, we know then from the spatial data that only the single breakpoint is the one that makes the most sense. And so all of this work then is built on data that researchers are producing. And so please help us, build these better models. Researchers, we need more thermal performance data to build these better functional forms. And then agencies, we need that spatial data from the field to try and validate these first principles models. And so that work then allows us to look at US establishment from first principles population dynamics models. We have published paper and an app. We've also looked at this establishment potential, thinking about what host lanternfly will eat as they spread across the US. And so we've seen some of these different studies. We have field surveys where they can look at what plant hosts lanternfly are feeding at in the invaded range. And then you have feeding trials where you can get really good estimates of host association, but only in a few key plants. And so both methods then have limits in forecasting the establishment of the pest on a wide range of novel hosts. You can't test everything that's out there that hasn't yet already been invaded. And so we know that closely related plants have similar traits and pest communities in general as we look across the tree of life. And so what my graduate student, Nick Huron, did was look at all the different data sets that are out there on spotted lanternfly host association and combine them in order to then build a model that explains those host associations by the phylogenetic relationships. So there's significant phylogenetic signal in host associations based on this data set. And then we can take that fitted model and project it out to, in our case, we looked at over 650 tree species across the US. And so now what we have then is a predicted estimate of host association across the United States. And then we built an app for that as well. And so you can zoom into your state, get a host list, and then get sort of that predicted potential impact of lanternfly if it moves then into your state. And so this estimate then is all available online. Another objective that our team has is to support pathway analyses. And so we do that by building pathway risk apps for states and basically asking this question of what properties, where are the properties that should be surveyed for spotted lanternfly? So we know that spotted lanternfly are spreading fast, and a lot of that is due to this jump dispersal events that's been occurring. They're being spread by humans. And so Victoria Ramirez, again, as an undergraduate, then helped me to build a pathway risk app for Pennsylvania. And so we worked with PA stakeholders to construct a pathway risk app, and they used this app. Then they found one of the outbreaks in Pittsburgh. And so what Nadège Bouillard has done, a postdoc in our group, she's then taken all of these locations of spotted lanternfly invasion hubs, these properties that could spread lanternfly, and then taken this large data set that I showed you at the very beginning of this talk, and then looked at the statistical association between jumps identified in that data set and how they're statistically associated with, how strongly they're associated with different infrastructure types, these different hub maps. And by doing that, then we can produce essentially a risk of a jump dispersal event at these different property types. And so if you have enough time and funding to survey all these different property types, then great, do that. But if not, then in this order, based on the statistical analysis, this is how you should be prioritizing different property types, surveying your landscape to see whether or not you get a jump dispersal event of spotted lanternfly. And so we built these different risk gaps for different states like California. And we're continuing to build more. And we've also produced then this code repository that allows any agency or any researcher to also build these risk gaps. And so all of that then is currently in prep, and then we have apps out. And then finally, our third objective of the research that we do is trying to effectively communicate spotted lanternfly risk. And so what I've shown you are a lot of apps. And so we have this dashboard that not only contains the apps that we built, but all these other tools that are out there for spotted lanternfly. And you can click on this link, slf.iecolab.org. And you'll get access to a lot of those apps that I'm showing you. And so there's this repository then of online spotted lanternfly resources that we've produced. And so for researchers and agencies, please share your apps and maps with us. We'll put them on the dashboard. It can reach a wider audience. And then agencies, if you want to share your public outreach and citizen science tools with us, we'll put those up there as well. So for these three objectives, what I've done is I've shown you various estimates of establishment potential based on both statistical and first principles modeling. These pathway risk gaps and an open source code framework that allows us to support pathway analyses. And then also this dashboard with all these apps of all the different deliverables. And so we very much take the approach that every model makes different assumptions. Just like what I showed you, where if you make these slightly different assumptions for one particular function in one complex model, you can get very, very different results. Those types of assumptions are being made in any type of model that you see. That you have to take many, many models and combine them in order to get accurate and precise management options and control. You have to combine all these different models in order to increase accuracy and precision. So thank you to all the funders and to all the senior personnel in the three labs that I told you about. And I'm happy to take any questions.
Okay, thanks, Matt. Yeah, there's some answers I think have already gone into the Q&A from other members of your lab. There is two in here asking if you could put the IECO website into the chat. That you missed part of it.
Sure.
And then the R0 map that you showed seemed like it was saying that SLF couldn't establish in parts of Pennsylvania where it currently is. Were they reading it correctly?
Yeah, so that R0 map shows that certain parts of Pennsylvania have a lower on average establishment potential than others. Some of those parts are below 1 and some of those parts are above 1. For those sort of on the edge of 1, you couldn't really see it in the map very well. But the answer is yes, there are parts of Pennsylvania and even New York that just based on this first principles model and the data set, the temperature data set that we're using, it's a slightly older temperature data set. It's saying that in the long term, lanternfly won't establish there. And so this type of modeling then essentially shows us that as you go further north, if there isn't sort of this climate change that I already talked to you about, then we do expect maybe some of these populations might be blipping in and blipping out. They might get recolonization events that occur. We, of course, don't exactly know what the truth is. And a lot of our modeling is based on a paucity of data as well, a paucity of experimental data on the relationship of temperature to development and mortality.
Okay, thanks, Matt. You can keep an eye on the questions and on the Q&A part and see if there might be some others coming in later that and ask you to answer.
Great, sounds
Thanks,
good.
Matt. Yeah.
Thanks.